Hello students. Today's topic Parkinson disease. Let's check out what is Parkinson, what it causes, and what is the pathophysiology for that. Let's check. In this slide, we will see introduction about the Parkinson, etiology, what are the causes for causing Parkinson, and its pathology, symptoms, clinical features, diagnosis, and the reference. And let's see. First of all, introduction. We see here it is a neurodegenerative disorders. In that category, Alzheimer's disease also comes and Parkinson's also comes. Let's see what is Parkinson. In 1817, James Parkinson, the scientist who first described this disease, firstly it named as shaking palsy. Afterward, on the name of the scientist, this disease get forwarded like a Parkinson disease or the Parkinsonism. Above age 65 year, 1% of population will have the Parkinson disease. The incidence of the Parkinson increases with the age, but an estimated 4% of people with Parkinson disease are diagnosed before 50 years. Men are 1.5 times more likely to have the Parkinson than women. What are the causes and what are the risk factors for causing the Parkinson's are idiopathic, which is the cause is not known up till now, viral inflammations, brain trauma, stroke, poisoning by manganese, carbon monoxide, pesticides, 1-methyl-4-phenyl, 1,2,3,6 tetrahydropyridine, MPTP, genetic predisposition, environmental toxins, environmental factors, drug induced Parkinsonism, age, genetics. We can see here we can see the pathophysiology or pathogenesis of Parkinson. We here are seeing Parkinson pathophysiology in different types. First is depend on risk factors like age, genetic disposition, environmental factors. By that risk factors, what happen? The mutation and the depositions of alpha synuclein. Then what is alpha synuclein? Alpha synuclein, it is the protein present in the brain. And smaller amount of alpha synuclein also present in heart, kidney, muscles, etc. Then after what? The induction of Levy bodies. What is Levy body? It is the deposition of alpha synuclein. A Levy body, the function of Levy body up till now not known, but it is present in the Parkinson disease, or the formation of Levy body, it is in the Parkinson disease. After that, Levy body depositions or the neuromelanin. What happens after that? After the Levy body formation or induction, microglossis occur. What is microglossis? It is the mitochondrial dysfunction which is found in the brain cells. After that, inflammations and generation of ROS. Then what is ROS? ROS is the reactive oxygen species, which is mitochondrial function. Other mitochondrial function is electron transport chain, which is coupled with reactive oxygen species. If we have to see reactive oxygen species, then we will need a next chapter or next topic we will see on the ROS. But for the students, you only understand what is ROS. ROS is the coupled reaction with electron transport chain formed by the mitochondria in the brain. Next, after that, distinguish different, there are two types. What will happen? First is oxidative damage to DNA. After what? After the generation of reactive oxygen species or mitochondrial dysfunction or the inflammation, oxidative damage to lipids, then oxidative damage to DNA, lipid, it will occur, and after that oxidative damage to the protein, and after that the loss of GSS. What is GSS? A GSS is a Gertzmann-Straussler shrinkers. It is responsible for neuronal functions. It is responsible for normal functioning of the brain. If the loss of GSS occur, then normal functioning of the brain or the neurons, it will not happen. It will cause a neurodegenerations. And on the other hand, alterations of the dopamine metabolism. And what is dopamine? It is the neurotransmitter, which is after that redox and axonal transport dysfunction will also occur. And after that, what will happen? The induction of apoptosis, which is the cell death, abnormal movement, and after that Parkinson's symptoms we can see in the patient by the risk factors, age, genetic disposition, and the environmental factors. Let's see the other pathophysiology of the Parkinson, the other way, that is free radicals and deficit in the energy metabolism. Here we are seeing what are free radicals. Free radicals, most common cellular free radicals are hydroxyl radical, that is OH, superoxide radical, that is O2, nitric oxide, NO. Other molecules such as hydrogen peroxide, that is H2O2, peroxynitrate, O and OO are not free radical but can lead to the generation of free radicals through the various chemical reactions. Here, dopamine as a relative unstable molecule in the nature which undergoes autooxidations, metabolism in the nigrostriatal tract system, thereby producing ROS. Here we can see if the free radical comes, which are discussed in the previous slide, here it will affect on protein, lipid, and DNA. What is the action of free radical on the protein is thiol oxidation. Then what is thiol oxidation? Thiol oxidation is the bromine iodine oxidation, is a organosulfur compound which is present, which is occur in the presence of bases. Next, lipids. What will happen here? Lipids, lipid peroxidation and membrane damage. And what happens on the DNA? Alteration gene expression and depletions of ATP and NADP. And after that, cell injury occurs or the inflammation occurs. And after that, ROS, which is reactive oxygen species, will increase. And here, after that, NFKB or NFR2, which is mediated gene expression, which regulates the gene expression, will dysfunction and neurodegenerations can occur. And after the neurodegeneration, we can see Parkinson disease. Other pathogenesis for the Parkinson is dopaminergic generation, program cell death. Here we are seeing dopaminergic generation in the substantia nigra may cause Parkinson disease. And see, what is substantia nigra? It is the basal ganglial structure located in the midbrain. A healthy cell. In the healthy cell, the caspase independent and caspase dependent. In caspase dependent, apoptosis occurs. In caspase independent, autophagic cell death occurs and the necroptosis occurs. Necrosis and the apoptosis both happen simultaneously. That's why necroptosis. What is caspase? Caspase are the family of proteins enzyme playing the essential role in the programmed cell death, which includes apoptosis, protosis, necroptosis, and the inflammation. After these all pathogenesis, we will see the clinical features. Clinical features we will see in the three forms, that is primary motor symptoms, secondary motor symptoms, and secondary motor symptoms. Primary motor symptoms and secondary motor symptoms. Here we see primary motor symptoms: resting tremor, which are shaking of hands, arms, or the legs, especially when the limbs is at rest. Next one is bradykinesia, gradual loss and slowly down spontaneous movement, rigidity, abnormal stiffness in a limb part of body, postural instability, impaired balance or the difficulties in standing, walking, gait and walking problem. Bradykinesia, postural inability both contributes to the walking and gait. Secondary motor symptoms: shuffling gait, microphagia, mask-like expressions, and speech difficulty. Secondary symptoms: dystonia, stoop posture, shuffling gait, reduced movement, akathisia, speech problem, reduced swallowing movement, microphagia. Here, all the symptoms and clinical features we see in the Parkinson's patients. Then, what are the diagnoses for that? Physical examination, MRI, CT scan, ultrasound, PET scan, which we can see here. Let's thank you.